Today, we're going to be talking about advantage and limitation of targeting GSK-3 in cancer therapy. This presentation is prepared by Jeremy, Jengi, Eldrian, and Azizul. The contents of the presentation include an introduction to GSK-3, advantages and potentials in targeting GSK-3 in cancer therapy, some examples, limitations of targeting GSK-3, the future directions of GSK-3 targeting, and the conclusion.
Now we start with introduction. So what is GSK-3? GSK-3 or glycogen synthase kinase 3 is a proline-directed serine threonine kinase that was initially identified as the enzyme responsible for phosphorylating and inactivating glycogen synthase, the key enzyme in glycogen biosynthesis. GSK-3 exists as two highly conserved isoforms in mammal, which is GSK-3 alpha and GSK-3 beta, both of which are ubiquitously expressed across tissue. GSK-3 beta is encoded by GSK-3b gene in human and is involved in energy metabolism, neuronal cell development, and body pattern formation. Move on to the next part, role of GSK-3 in normal cell and cancer cell. For role in normal cell, GSK-3 regulates glycogen synthesis, glycogen metabolism, and insulin signaling. Moreover, it can control cell cycle, progression, and cell fat decision. It also can modulate programmed cell death and autophagic process. For role in cancer cell, aberrant regulation of GSK-3 beta disrupts cellular activity including proliferation, differentiation, and apoptosis, driving tumorigenesis. GSK-3 is co-opted in cancer for tumor growth, fibrosis, and immune evasion. GSK-3 beta invasion can enhance chemotherapy cytotoxicity while simultaneously modulating anti-tumor immune response. So now let's move on to major pathway related to GSK-3. First, we have WNT beta-catenin pathway. GSK-3 acts as negative regulator. GSK-3 phosphorylate beta-catenin targeting it for degradation in the destruction complex. Next, we have PKB pathway. Protein kinase B phosphorylate and inactive GSK-3. GSK-3 downregulation correlate with PI3K oncogenic pathway activation. Last but not least, we have nuclear factor kappa-like chain enhancer of activated T cell. GSK-3 positively regulate NFKB-mediated transcription, inhibition, downregulate NFKB, and decrease survival genes. For GSK-3 significance in cancer, first, when GSK-3 is inhibited, beta-catenin accumulate and translocate to the nucleus, activating oncogenic transcription such as cyclin D1 and CMYC. Next, protein kinase B mediated GSK-3 inhibition promotes cell survival and proliferation, such as non-steroidal anti-inflammatory drugs. Chemo prevention work by upregulating GSK-3 beta while downregulating PI3K ARC. Last but not least, NFKB activation promote inflammation, immune evasion, and tumor survival. Elraglusib decrease NFKB expression.
Now we will look into the advantages and potential of targeting GSK-3 in cancer therapy. To understand a little bit more about this topic, we will look into three main questions. First, why can targeting GSK-3 help in cancer therapy? Second is, how can the inhibition of GSK-3 help to reduce cancer cell growth or survival? And can GSK-3 inhibitor actually improve chemotherapy and radiotherapy response? So, why can targeting GSK-3 help in cancer therapy? Well, a variant of GSK-3 known as the GSK-3 beta is an active constituent enzyme in the cytoplasm of resting cell. So, when it is phosphorylated and activated, it will accumulate in the nucleus via translocation and the mitochondria. This will activate a lot of downstream transcription factor and recruits even more transcription factor. To note that there are more than 40 proteins can be downregulated by GSK-3 beta. This makes inhibition of GSK-3 beta important because we can regulate a lot of downstream processes. GSK-3 beta is actually known as a tumor suppressor where it can reduce cancer cell survival and growth. It can stop tumor growth and proliferation through several mechanisms. For example, GSK-3 beta can target and degrade several pro-oncogenic proteins and cell cycle regulators, such as cyclin D1, P, and P53, to trigger their destruction and preventing them from entering the nucleus to kickstart a cell cycle and arresting the cell cycle. It also can mark B-catenin for destruction in order to inactivate pro-survival PI3K AKT pathway. Also, blocking of GSK-3 will inhibit nuclear factor kappa B transcriptional binding in order to downregulate anti-apoptotic factor to trigger intrinsic apoptosis. Speaking of apoptosis, targeting GSK-3 beta can also increase the mitochondrial membrane potential to forcefully produce reactive oxygen species to trigger apoptosis in the tumor cell. One of the advantages of using GSK-3 inhibitor in targeting GSK-3 is that it can improve chemotherapy or radiotherapy. For example, GSK-3 beta inhibitor along with chemotherapy downregulates GSK-3 beta, which causes increase of therapeutic effect of 5-fluorouracil to abolish cell viability, colony growth, reduce xenograft tumor mass in various drug-resistant P53 null colon cancer line. Also, GSK-3 beta inhibition allows for chemo-resistant carcinoma cell to become more susceptible to the drugs such as sorafenib, which helps to inhibit cell growth and angiogenesis. GSK-3 beta inhibition also enhances the sensitivity of glioblastoma cell to radiation to improve the therapeutic effects of temozolomide. Finally, GSK-3 beta inhibitor enhance the sensitivity of pancreatic cancer to gemcitabine by decreasing the expression of tumor protein 53-induced nuclear protein 1 or double-stranded repair-mediated P53 target gene, and by downregulating the CDK4/6 complex-dependent phosphorylation of RB tumor suppressor protein.
Next, I would like to share some examples of GSK-3 beta inhibitors that are used in current cancer therapy. The first is tidilgusib, which is a non-ATP competitive GSK-3 beta inhibitor used in the treatment of glioblastoma in patient-derived xenograft in mice. Secondly, we have AR-A014418, an ATP competitive GSK-3 beta inhibitor, which has shown efficacy in treatments of gastric cancer, synovial sarcoma, and fibrosarcoma. It functions by suppressing the proliferation of cancer cells and inducing apoptosis. AR-A014418 has been tested both in vitro and in vivo studies. The third GSK-3 beta inhibitor is TWS-119, a non-ATP competitive GSK-3 beta inhibitor used to treat human alveolar rhabdomyosarcoma and triple-negative breast cancer. In human alveolar rhabdomyosarcoma, it helps to regulate the epithelial mesenchymal transition, EMT, while it helps to regulate cancer stem cells in TNBC. Finally, we have 9-ING41, which is the most promising GSK-3 beta inhibitor currently because it is in clinical trials as a single and combination agent. It has been shown to be useful against neuroblastoma, bladder cancer, and glioblastoma, both in vitro and in vivo studies.
Now, moving on to the limitations of inhibiting GSK-3. So GSK-3 inhibition is promising, but there are limits to what we can do. So I've broken them down into three main ones. The first one is side effects and concerns. So are there any safety concerns when we're doing GSK-3 inhibition? And tumor promoter or suppressor? meaning GSK-3 can act as tumor promoter or tumor suppressor. So does it mean that when we inhibit GSK-3, is it always beneficial? And finally, targeting difficulties. Because not every cancer that happens is based off GSK-3 production. So is the target always the same for cancer? What if the tumor is not dependent on GSK-3? So we'll look into these three limitations. So the first limitation of inhibiting GSK-3 beta is because of its side effects and its concerns. So GSK-3 beta actually controls 5 major pathways inside our body. The first one is metabolism, the second one is inflammation, the third one is cell survival, fourth one is apoptosis, and the fifth one is our immune response. So when we inhibit GSK-3 beta, there will be different effects happening to each of these pathways as shown here. So for example, in metabolism, when GSK-3 beta is inhibited, it will increase glycogen synthesis. So our glucose homeostasis will be disturbed. And then for inflammation, because it's involved in the NF-kappa beta signaling, if GSK-3 is inhibited, this will reduce our inflammatory response. For cell survival, because GSK-3 beta regulates cell survival signaling, this will change the survival pathway balance and cause cellular dysregulation. Furthermore, for apoptosis, because GSK-3 beta promotes mitochondrial apoptosis, if we inhibit GSK-3, it will reduce apoptosis. So this means that tumor cells will be allowed to survive. Then finally, immune response. So because GSK-3 beta regulates cytokine production, if it's inhibited, there will be an imbalance in cytokine, and this will disrupt our immune system homeostasis. Now, on to the second limitation. So GSK-3 beta actually can act as a tumor promoter or tumor suppressor. So as a tumor promoter, the active GSK-3 beta will trigger the NF-kappa beta signaling. So this pathway will activate pro-survival and anti-apoptotic genes such as BSL2 and survivin. So this actually promotes cancer cell survival because the cancer does not undergo apoptosis and is allowed to proliferate and might migrate or invade other organs. So this will cause an even more aggressive cancer growth. However, GSK-3 beta can also act as a tumor suppressor. As mentioned by Eldrian earlier, active GSK-3 beta can phosphorylate beta-catenin. So the phosphorylated beta-catenin is degraded. So this prevents beta-catenin from accumulating in the nucleus. So this will prevent or reduce transcription of oncogenes such as cyclin D1 and MMPs. So this will actually stop cancer cell proliferation. So as you can see, GSK-3 can act as a promoter or suppressor. So by inhibiting, we might be inhibiting its tumor promoting properties, but at the same time, we will also be stopping its tumor suppressing properties. Another important limitation of inhibiting GSK-3 beta is targeting difficulties. So as we all know, not all tumors will actually rely on GSK-3 beta. There may be other mutations that cause tumorigenesis in the first place. So response to GSK-3 beta inhibitors may vary between patients because each patient will have their own tumor type. So it is better for us to do biomarker testing so that we identify which patient is suitable for GSK-3 beta inhibition therapy. So this will reduce unnecessary toxicity to the non-responders. So basically, we have to biopsy the cancer cell. Then I can do GSK-3 beta marker tests either via Western blot, sequencing, or biomarker blood testing. So then we can identify which patient is suitable for this therapy and which patient is not. So this will actually prevent unnecessary toxicity or damage, especially to the cancer patients. So back to the main slide, GSK-3 beta inhibition is promising but with limits. So we can see here there are three main limitations, and all of them are talking about how GSK-3 beta may actually affect other pathways, or the tumor cell in question will not be affected by GSK-3 beta in the first place. So it's better to know the problem or know what pathway the tumor is using first before we inhibit. So this will actually prevent unnecessary pain or errors, especially when treating the patient. Thank you.
For future directions in GSK-3 beta, scientists are looking at immunotherapy, more specifically NK cell activations. NK cells are the body's natural defense against tumor cells. They have NK receptors such as NKGD2, MIC-A and MIC-B, and ULPB. GSK-3 beta normally activates the STAT3 pathway, which inhibits the upregulation of MIC-A and MIC-B NK receptors. By using small GSK-3 beta inhibitors, we aim to inhibit GSK-3 beta and therefore inactivating STAT3 pathway, which allows the NK cells to upregulate the MIC-A and MIC-B receptors.
So, in short, when it comes to advantages in targeting GSK3, because GSK3 is linked to cancer pathways, targeting it will reduce tumor growth and survival. So, GSK3 inhibitors will improve chemotherapy and radiotherapy responses. In current research, GSK3 inhibitors such as 9-ING41 and elrabglucid, they are still being studied in cancer therapy. They are quite promising, but still they are not standardized for approved cancer treatment. When it comes to the limitations, because GSK3 can act as both tumor promoter and tumor suppressor, it is best to understand which type of cancer does the patient have before we target GSK3. So patient selection and biomarker testing is very important. Finally, when it comes to future directions, currently they are trying to develop safer and more selective inhibitors. So they will use biomarkers to choose suitable patients first. Then only they combine GSK3 inhibitors with chemo, radio, or immunotherapy for better performance and better chances of recovery. Thank you. These are our references used to prepare this presentation.